Human locomotion and movement result from the contraction and relaxation of skeletal muscles. Muscles are attached to the bones by tendons, tough bands of fibrous connective tissue. Muscles are covered with epimysium, a layer of connective tissue. Muscles are composed of fascicles, which are covered with perimysium, another layer of connective tissue. Fascicles are composed of muscle fibers. Muscle fibers are formed by the fusion of muscle cells, and they can therefore be considered single multinuclear cells. They contain actin and myosin, proteins that produce contractions in the muscle. Actin and myosin are bundled in myofibrils, which have a characteristic striated structure due to the organization of the actin and myosin filaments. This is why skeletal muscles are also called striated muscles. The band that contains myosin is called the A band. The H zone, located within the A band, only contains myosin, while the I band only contains actin. When the muscle is stimulated, the actin and myosin filaments slide over one another. The I band and the H zone become shorter, and the muscle contracts. The molecular mechanism of muscle contraction is as follows. Stimulation of the muscle results in an increase in the calcium level of muscle fibers. Calcium ions bind to the troponin, which causes tropomyosin to move, and thus the myosin-binding parts of the actin become uncovered. Myosin binds to the actin, ADP and phosphate are released, and the myosin head bends and pushes the actin. Then ATP binds to the myosin head, which releases the actin. The ATP molecule splits into ADP and phosphate, and the myosin binds to the actin again. This process continues while the calcium level in the muscle fiber is high. When the stimulation is over, the calcium level decreases, and the troponin-tropomyosin complex returns to the default state.